In progress.
Hello, hi everyone. My name is Dr. Sana Khan, your biology expert, and in this video, we are going to talk about antigen-antibody interaction. I hope you have seen the previous video where we have already talked about the structure of antibody. Correct? It was very easy, right? Do you remember it? It was 4 polypeptide chain held by disulfide bond, right? One end was amino group, the other was carboxyl group. Correct? Then there was a point where the arms and the stem were getting attached, that was called as hinge. And there were 2 heavy chains, there were 2 light chains. Each chain was having 2 regions, that is the variable region and constant region. Variable region keeps on varying. Why? Because it has a paratope. What is paratope? It is a antigen binding site, right? And then the constant region is going to be constant. And then we talked about how the antigen and antibody interact with each other. They interact in which manner? In lock and key mechanism. And there was one more word which we did, and I told you that you should not forget it, and that is epitope, which is the antigenic determinant. Okay, so now in this video, what we are going to study is how the antigen and antibody interact. So now we know that once the antigen goes inside, antibody and antigen interact with each other. Now, how do they do? Let us understand in this video. Okay. So, antigen-antibody interaction includes 3 most important types. Okay. So, it includes 3 most important types of interaction. The first one is called as agglutination. What is it called? Agglutination. The second one is called as opsonization. क्या कहते हैं? Opsonization. And the third one is called as neutralization. What is it called? Neutralization. So what are the 3 interactions? Let us revise. Agglutination, opsonization, neutralization. Once again, agglutination, opsonization, neutralization. Now let us understand each of the interaction in detail. It's a very nice video. I think you're going to enjoy it. See, first we're going to talk about agglutination. So pathogens have come. Can you see? Pathogens have come inside. Okay, they're dancing inside our body. But antibody, but the macrophages in our body is very alert. As soon as the antigen goes inside our body, macrophages get very, very active. And now what does this macrophages do? Macrophages will engulf this pathogen one by one. See, macrophage will engulf this pathogen one by one. But don't you think it's a strenuous process, right? Engulfing it one by one, one by one. Instead of that, what does the antibodies do? They come and the free antibodies, they come and they glue all the microorganisms together. What do? They cause agglutination. That is, they glue all the microorganisms together. Okay, because once that lot of pathogen comes inside, the macrophage has to eat them one by one, which becomes difficult. So to avoid that, what does our free antibodies do? The free antibodies glue all the pathogen. What does it do? It glue all the pathogen, and that is called as agglutination. And now it becomes easy for the macrophage to engulf this immobilized mass, because when all the antigens get glued with the antibodies, it becomes an immobilized mass. They are not able to move because they are formed in a group. They are glued together. And now this macrophage will engulf this immobilized mass. Can you see? Now this macrophage is going to engulf that immobilized mass. Okay, and that is called as agglutination. What is it called? Agglutination. So when the free antibodies glue all the pathogen together so that the macrophage can eat them together, that is called as agglutination. So agglutination of bacteria and viruses can occur in our body by the free antibodies present in our body. Now let us go to the second one, that is opsonization. So what does the word opson means? Opson means delicious side dish of any meal. Okay, it's a delicious side dish of any meal. Now the pathogen has come inside. This is the macrophage. Macrophage has understood that the pathogen has come inside. So macrophage normally will eat this pathogen. What does the macrophage will do? Will eat the pathogen. But it is a non-specific response. Why? Because here the antibodies have not come. Yes or no? Antibodies have not come. So to avoid that non-specific response, what does antibodies do? Antibodies combines with the pathogen. Can you see? Antibodies combine or get coated with the bacteria. So whatever free antibodies are present, they are going to come in and they are going to coat this bacteria, and now it will become a specific response. And now the antibody coated bacteria is going to be engulfed by the macrophages. Can you see how is it engulfing? So finally it will be phagocytized. Is it clear? And that is called as opsonization. So in agglutination, many bacteria which were moving were made into a mass which was immobilized, and then macrophage consumed it. Okay, phagocytosis. But in opsonization, to avoid the non-specific response, the pathogen gets coated with the antibody, and then finally the bacteria will, the macrophage will engulf that antibody-coated bacteria, and that is called as opsonization. Is it clear? Now, coming to the last one, that is neutralization. So now certain pathogens release toxins, right? So there are certain pathogens which will release toxin, and that will get attached to the receptor of our body cell. Can you see? See, pathogens have released toxin. This is our body cell, and finally the toxin has got attached to the body cell, and it damages our body cell. It damages our body cell. This is the normal mechanism. So whenever a bacteria comes in, there are certain bacteria which will release toxin. That toxin will bind to our cell and it will damage. So now what happens when there are antibodies circulating in our body? So now the free antibodies, what does they do? They start protecting our cell by combining with that toxins. Can you see? So they are stopping the toxin and they are not letting the toxin to reach our body cell. So they prevent binding of toxins to receptor, and that is called as neutralization, because they neutralize the effect of that toxin and our body cells get protected. Understood? So here this example is for tetanus toxin, because even it releases the toxin and damages our body cell. Is it clear? So there are three types of interaction. The first one was agglutination, second is opsonization, and third is neutralization. So agglutination, when many bacteria are moving, our macrophage will have to engulf each one of them, which becomes difficult. So the free antibodies get coated with the bacteria, and now the macrophage can eat up that immobilized mass. Opsonization, to avoid a non-specific response, free antibodies get bind up with that pathogen, and now the macrophage will engulf it. And last one is neutralization, where the certain toxins will get released due to bacteria, and that will bind to our body cell and it will damage it. So to prevent that, free antibodies bind with that toxin and doesn't allow our body cell to get affected. Is it clear? So these are the three most important interactions which an antibodies show. Okay, so that's all about this video. I will soon meet you with the next video. Till then, bye, take care and work hard. Bye.